Today we are going to talk about the cell theory and the cellular components. First of all, I describe that what cell theory states. Cell theory states that all living organisms are made up of one or more different types of cells. Second part, it states that cells are the basic unit of life. The third point is that all the cells arise from the pre-existing cells. That is, all the newer cells are formed by divisions in the older cells. Now we look at it, how this theory developed. Cells were first described by Robert Hooke, was the curator of instruments of the Royal Society of London. In 1656, he made a simple type of microscope by using different types of lenses to magnify different things. He observed in the thin slices of cork some sort of cells. Now we know that he observed certain sort of dead cells. Later on, another person who was actually a textile merchant, he also developed magnifying lenses, and by the setup, he also made a sort of microscope. With the help of that microscope, he observed different types of single cellular, or we can say unicellular organisms in the pound's water. He was Leeuwenhoek. So Leeuwenhoek observed actually the unicellular organisms, and Robert Hooke observed the plant cells. After that, in about 1809, about 200 years later, there was a person, Lamarck. He proposed that a body is living if it consists of cells. There's another person came later, Robert Brown. He identified nucleus inside the cell. He says that cell have central part that is more dense in comparison to the other parts of cell. In 1838, a botanist named Sheldon, he actually stated the first part of the cell theory, actually half part, that all the plant cells, all the plants, the multicellular plants, they are aggregates of cells. Later on, another cytologist, and more precisely a zoologist, an animal scientist, Chauvin, stated that all animals also consist of cells. These two theories combined. Various other people actually looked at these, and they combined to make the modern form of first part of cell theory. That is, all organisms, the living organisms, consist of one or more cells. In 1855, another scientist, Virtue, described that cells only arises from pre-existing cells. That is, if there are some cells which are present, they divide and they produce the new cells. Pasteur later on produced the proof for this theory, that is, cells arise from pre-existing cells. In this way, the cell theory took its current form, that is, all the living organisms consist of one or more cells. Cells are the basic unit of life, and all cells arise from the pre-existing cells. This is about important features of cell theory. Now we are going to do about cellular components. Cell consists of many components. Cell is covered, all of the cells are covered by a membrane called plasma membrane or the cell membrane. This membrane protects the internal parts of the cell. Plant cells, fungal cells, and the prokaryotic cells, they also have another layer outside the cell membrane, which is called cell wall. So plant cells, the fungal cells, and the prokaryotic cells do have a cell wall that is external to their cell membrane. Cell wall is even a more protective layer. It's more hard in comparison to the cell membrane, and in part, sometimes in few cells, it's like that. Inside the membrane, there is a plasma, a fluid, that is called cytoplasm. Cyto of cell, plasm, fluid. Cytoplasm is the fluid that is present between the cell membrane and the nucleus. Cytoplasm have many components. All the organelles are there, all the functions like metabolism performed there, and so on. Cell also have its skeleton, a framework which gives cells its shape, help it in its movement, and set its different organelles on their place. Then comes the organelles. cells have organelles: mitochondria, nucleus itself, endoplasmic reticulum, Golgi bodies, vacuoles, and a lot many more. Here you can see a diagram of an animal cell, which shows that it have a cell membrane outside, then inside, almost in the center, you can see a nucleus, then you can see surrounded nucleus, the endoplasmic reticula, a network. After that, you can observe the Golgi complex, few mitochondria, centrioles, cytoplasm, not many other structures. Now we go ahead and look at these structures one by one. The cell membrane or the plasma membrane, it covers all the components of cell. This actually gives a cell, individual cell, a state of integrity, or we can say that it encloses the contents of the cell in a specific environment. What are the functions of plasma, or the cell membrane and the plasma membrane? It acts as a barrier, it gives protection to the cell, and it allows transport of materials. The cell membrane act as a barrier. Cell membrane is semi-permeable. That is, it allows few molecules to pass in or go out. It does not allow some other molecules to move in or move out. So it means that it acts as a barrier between the internal part of the cell and the external environment. It gives protection to the internal parts of the cell. Internal parts of the cell are delicate. They may damage in response to any pressure or tension exerted on the cell. Cell membrane protects it, separate or isolate the inner part from external environment. It provides transport of materials. Cells need many materials that should come in or should go out on intervals. Cell membrane or the plasma membrane is semi-permeable. It manages the transport of materials which are required, like food particles which are required by the cell. It allows food particles to come in. It allows the waste products to go out in a very technical way. For example, cells need water. Cell membrane, by specific channels in it, allows those water molecules to move in or move out. Now look at a diagram. This diagram shows you different parts of plasma membrane or a cell membrane. This diagram shows that the cell membrane, plasma membrane, as it is generally said, is a fluid mosaic. It is a fluid because it consists of a double layer of lipid molecules, the fat molecules. These fat molecules, as you observe, have their heads on one side and have their tails in the inner side. On upper side, the top, you can see the extracellular part. On the bottom, you can see the intracellular part, that is inside of the cell. Cell membrane consists of a double layer of lipids, which give it a fluidity. It's like a fluid, but it also have various proteins integrated. As you can see in the diagram again, there are different types of proteins which are integrated at different places. For example, if we start from the left, we can see two large proteins which are close together. Both are crossing the whole of the fluid bilayer, that is the lipid bilayer. Then you can see a small protein which is present down there towards intracellular side, labeled as peripheral protein. Peripheral is present on the periphery. It is present towards the inside. Below that, you can see rope-like structures which are attached to these peripheral proteins. These rope-like structures are the cytoskeletal elements. These elements attaches to the peripheral protein, and on the other side, they attaches to different organelles. For example, mitochondria, nucleus, maybe some else, endoplasmic reticulum, and they give support to those organelles because, cytoplasm is a fluid. It's a plasma. Organelles have to be placed have to be kept at a particular location, at least for some time. And they are placed on these locations with the help of these cytoskeletal elements. And then the cytoskeletal elements, the fibers, the ropes, as you can see, they are attached to the peripheral proteins. So cell membrane gives support to the organelles with the help of these cytoskeletal elements. You can see various different types of proteins above, to which certain carbohydrate groups are attached. We call them glycoproteins. Glyco are carbohydrates. Proteins which are attached with carbohydrate groups. Most of these proteins act as receptors. That is, when any molecule have to come in and give some message to that particular cell, they have to come and attach to a glycoprotein many times. So glycoproteins, they receive the signals. They are receptors of the cell. Just like that, you can see that integrated inside the cell membrane in its yellower part towards the tail part, there are cholesterol molecules. This lipid bilayer is like a fluid. It needs some support. It should be supported by some more strong structure. These cholesterol molecules, they give support and strength to the cell membrane. Now, in different types of cell membranes, the concentration of these cholesterol molecules, that is, quantity of these cholesterol molecules, may be more to less. If membrane needs more strength, then there are more cholesterol molecule. If it needs less strength, there are less cholesterol molecule, depending upon in which environment a particular cell is living. You can see certain glycolipids. There are certain carbohydrate groups which are attached to the surface of the lipid bilayer. Because they are attached to the lipid, they are called glycolipid. Glyco for carbohydrate, lipids for the fat molecules. This is quite about structure of the cell membrane. So, cell membrane is a fluid mosaic. It allows transport of materials inside and outside, and it acts as a protective barrier. Now we talk about the movements across the cell membrane. This is very important, actually one of the most important features of the cell membrane, that it is semi-permeable. It allows certain molecules to pass. It does not allow other molecules to pass. And also, this actually very technically separate the internal and external environments. There are certain molecules which are lipid-like. As we know from the structure that the cell membrane overall, so if a molecule is like a lipid, for example, there is a fat type hormone, it can directly pass through the cell membrane and come inside, or it can pass out, but the other things, the other molecules like proteins, like carbohydrates, they cannot pass through, like ions, water. They cannot pass through directly through the cell membrane because it's a fluid, it's a lipid, and lipids they actually repel those molecules. So what happened that for movement of this type of molecules, cell membrane have channels, the ways from which these molecules can pass, and these channels, these ways, are made up of proteins. These proteins are very, very specific in their structure. One type of channel usually allows only one type of molecule or ion to pass. For example, there are channels called water channels or aquaporins. Aqua for water, porins, pores. So these are the protein channels made up of proteins present integrated inside the cell membrane, which allows only water molecules to pass. There are certain other channels. For example, sodium ion is a very important ion present inside the cells and in their extracellular environment. Sodium ion have particular channels through which it can pass. We call them sodium channels. Just like that, there are protein channels, there are calcium channels, there are potassium channels, and so on. And these channels are not, many of these channels are not always open. There are few channels which are always open. We call them leak channels. From these channels, ions or molecules can pass against a concentration gradient. For example, if they are more outside, they will move towards inside. If they are more inside, they will move towards outside. But there are other channels which allow only a specific type of molecule to pass, and they open and close as required by the cell. Certain signals come and tell these channels to open, or other signal comes and tell these channels to close. So these are open and close upon the requirement of the cell. For example, during nerve impulse, when a message from brain is transmitted, for example, to the muscles, or message from muscle is going to the brain, there are sodium channels which need to be open. And these are opened with the help of a stimulus, that is, some signal. When a stimulus comes, a signal comes, these channels open and allows the sodium to move in, and the cell transmit a message. Here you can see a diagram. In the diagram that you can see on your screen right now, it can show you various types of channels. Few on your left are open protein channels. Few on the other side shows when they are closed. So channels open and close. And how they open and close? They open and close by a conformational change. I hope you understand the conformational change, but I simply describe it. Conformational change is this, that arrangement of molecules of a macromolecule, or arrangement of atoms of a molecule in space is changed. This is called a conformational change. For example, if they were towards right, they move towards left. With the result, channel open or close. So this was about the cell membrane, that what is cell membrane and how it works. Now we talk about the cell wall. All the plant cells, the algae, fungi, and the prokaryotic cells do have a cell wall. Cell wall is a layer, consists of different types of material which is present outside the cell membrane. The plant cell wall consists of a specific fiber, long fiber, called cellulose. It also have certain other fibers included in it which attach different cellulose fibers to each other. These are called hemicellulose and pectin. The fungal cell walls consist of another type of polymer called chitin. In contrast, the cell walls of the prokaryotes, they consist of another fiber which is a polymer of the amino acids, different types of amino acids and sugars. This is called peptidoglycan. Now again, look at the diagram. This diagram shows structure of cell wall. Now the plant cell wall. Plant cell wall is of two types, primary cell wall and a secondary cell wall. The primary cell wall is present in most of those cells which have to perform like various functions, and sometimes the cells which are not yet mature. Some cells which have to bear tension, or for example the cells which have to allow the transport of water from roots to the stem or other parts of the plant, those cells have another cell wall, secrete actually another type of cell wall towards inside, which is called a secondary cell wall. So it means that primary cell wall remains the externalmost layer, then comes the secondary cell wall if it is present, then comes the cell membrane, then cytoplasm, and then the nucleus. Here in the diagram, you can see a structure of a plant cell wall, which is the primary cell wall. You can see the middle lamella, which is a layer present between the cells. Then you can see long structures in blue, big fibers. These fibers are called the cellulose microfibrils. You can see that they are arranged differently. There is one layer which is going horizontally at a straight angle. There is second layer which is going in comparison to the lower layer, and there is a third layer which is again in the same arrangement. Then you can see different cross linkings in between these cells, these microfibrils. These microfibrils are attached to each other with the help of certain fibers which are cross linkings. We call them cross linkings because these are crossing layers of the cellulose molecules, cellulose microfibrils. Then you can see there is a third molecule called pectin. Pectin is also attached at a different angle to the cellulose microfibrils. Pectin is a material that is present in the jams and jellies, and it is used to solidify those or convert them into a gel. What actually pectin do? It attracts water molecules and calcium molecules and some other, and convert this interior of the cell wall, the primary cell wall, into a gel-like matrix. Down there, you can see the plasma membrane. So it means that plasma membrane, then there is a thick layer of cell wall, the primary cell wall. Then comes the contact between the cells, the middle lamella. What are the functions of the cell wall? Functions of the cell wall is protection, strength, and support. Plant cells have these two types of cell walls. Primary cell wall give them support. Secondary cell wall give them even more support. The primary cell wall and secondary cell wall are different in their structure. We look at next diagram. Here you can see in that diagram that the lower layer, the layer which is external most, is the primary cell wall. Then comes different layers of the secondary cell wall. Secondary cell wall, you can see the outer layer, the middle layer, and the inner layer. These makes the whole of secondary cell wall. It means that secondary cell wall is secreted in layers. When a cell matures, initially it have a primary cell wall. When it matures, it secretes outer layer of the wall. Then if it needs more, it secretes more layer, and that's it. These layers, the difference between the primary cell wall and the secondary cell wall is this, that particularly primary cell wall have cellulose fibrils in comparatively a lesser quantity. The secondary cell wall have cellulose fibrils in a more quantity. They have more cellulose microfibrils present in them. The primary cell wall have more number of pectin and hemicellulose molecules in comparison. The secondary cell wall have less hemicellulose and pectin molecules. Secondary cell wall also have another characteristic, that is its cellulose microfilaments are more arranged. They are, in comparison to the primary cell, they are more arranged. That is, they are more ordered. And as you know, that if in a particular structure it is less ordered, it will have less strength. If it is more ordered, it have more strength. So in primary cell wall, cellulose fibrils, microfibrils, which give it more strength, they are less orderly arranged. They are less organized. In a secondary cell wall, they are more orderly arranged and they are more organized. With the result, they give the cell more strength. And secondly, as you can observe in the diagram, that these cellulose microfilaments, these are present in different directions. In the first one, you can see these are horizontally arranged. In the second one, these are vertically arranged. In the third one, they are arranged at another angle. This difference in arrangement gives more and more strength to the structure of the cell. So we finished talking with the cell wall and the cell membrane, the layers, the coverings. Now we talk about the cytoplasm. Cyto of cell, plasma, or the fluid. Cytoplasm is the living matter, part of the protoplast. This is a semi-transparent substance that have different types of materials, proteins, various carbohydrates, enzymes, and other materials present inside it in dissolved form or a semi-dissolved form. It means that these materials are present inside the cell, inside the cytoplasm, either in dissolved form or a semi-dissolved form. We can say it have organic material inside, it have inorganic material inside, which are dissolved in a watery fluid. The major fluid is water, and it have dissolved particulate matter that consists of organic molecules, proteins, lipids, carbohydrates, etc. And it have inorganic molecules like different types of like magnesium, calcium, potassium or so. What are the functions of cytoplasm? Functions of cytoplasm are, there are two major functions. One is that cytoplasm provide space for different types of organelles. All the organelles, mitochondria, the ribosomes, the endoplasmic reticulum, the Golgi bodies, they are all present inside the cytoplasm. Cytoplasm also provide a watery fluid which is required for different metabolic processes. It means that cytoplasm support and help in different types of metabolic processes. For example, the glycolysis, which is the breakdown of glucose, it occur in the cytoplasm, and enzymes which are required to break down glucose into smaller parts, different small parts, and to release energy, they are present inside the cytoplasm. They are dissolved in the cytoplasm. So cytoplasm gives a space for organelles, it gives a space for metabolic reactions to occur, and it gives space to nucleus inside as well. Now we are going to talk about the cytoskeleton, the skeleton, the cell. Cyto of the cell, skeleton, the framework that supports the cell. Cytoskeleton is the system that gives support to the cell and help the cell in its own movements. It also helps different organelles inside the cell to move from one place to another place when this is required. Cytoskeleton consists of three types of fibers. One is called microtubules, the other is called microfilaments, the third is called intermediate filaments. We talk about them one by one. The microtubules, these are the largest in diameter, and microtubules are hollow from inside. Their generalized diameter is about 20 nanometers. But remember, then when we are talking about the size of different cytoskeletal elements, we are only referring or talking about their diameter and not their length, because lengths of these fibers are different from each other, but their diameter is, their size actually refer to only their diameter. So microtubules in comparison to other are the largest. They have about 20 nanometer diameter, but they are hollow from the inside. They consist of a protein that is called tubulin. Tubulin molecules are of two types, alpha tubulin and beta tubulin. They join together to make the tubulin dimer. Dimer means that it is made up of two tubulin molecules. These dimers later on join together to make long hollow tubes called microtubules. The other one is called microfilament. These are the smallest among cytoskeletal elements. Microfilaments are about 7 nanometers in their diameter, and they consist of a protein called actin. Actin molecules are arranged in the form of beads, and there are two bead-based chains wound around each other to make a helix. We call it an actin subunit. The diameter of this subunit is 7 nanometers. Again, their lengths may vary. Then comes the third. These are called intermediate filaments. Intermediate filaments consist of a particular family of protein called the keratins. Keratins are the same proteins which make our nails, for example, the harder parts. It means these are tough proteins. The keratin family of proteins, different types of keratins, join to make intermediate filaments. It specifies their function. They give support. Though there are, now we talk about the functions of these cytoskeletal elements, or we can call them cytoskeletal fibers. Cytoskeletal elements, they have one major function, that is giving shape to the cell, giving support to the cell, support to its different organelles, keep them in place, and movement. We have a look on the diagrams which shows structures, generalized structures of these cytoskeletal elements. Here in the diagram, it shows you the structure of a microtubule. Microtubule, as I described previously, consists of tubule in diameter, and then these diameters are arranged in a circular fashion to make a long hollow tube. Length of these tubes depends upon how and which part of cell require them, for how long they are required. For example, if cell is using these microtubules for transport of an organelle, say a mitochondria, from one place to another, then the length of microtubule will depend upon the distance from the point 1, from where mitochondria have to be taken up, and till point 2, where mitochondria have to be placed. In the second diagram, you can see microfilament. Microfilament is an actin subunit, as you can see. It's a helical chain. In the third diagram, you can see the structure of an intermediate filament on the right. Their diameter is about 10 nanometers, and they consist of fibrous proteins, which initially their monomers, that is, smallest molecules, join together to make a dimer. Dimers join together to make tetramers. Tetra means 4. And these tetramers then join to make sheets. And these sheets are wrapped around each other, as you can see in the diagram, to make a thick and tough fiber, which is called intermediate filament. Now look at the diagrams which are colored. These are the pictures. If we want to know that in a cell, where at which places the cytoskeleton is present, we can stain them, because you know that the microtubule consists of tubulin, which is a protein. Microfilament consists of another protein called actin. And the intermediate filaments also consist of various proteins. So if we stain our cells, we give them a color by using anti-tubulin antibody, anti-actin antibody, or anti-keratin antibody, then we can look at them that where these elements are present. The pictures above, which are not diagrams but pictures taken through fluorescent microscopy, shows that cytoskeletal elements are present almost everywhere in the cell. The first diagram shows that in blue color you can see the nucleus. In green color surrounding them are microtubules. In the second picture, you can see that we stained this cell with anti-actin antibody, and it shows that in almost all part of the cells, these actin filaments are spreaded. So it means that cytoskeletal elements are present almost everywhere in the cell and support all parts of the cell. In the third diagrams, you can again see inside nucleus in orange color, and in green color different types of intermediate filaments. This is another picture taken more closely, in which you can see various cells which have nuclei in blue color, and in green color they have microtubules, and in red color they have actin. That is, these are stained for microtubules, actins, intermediate filaments, all of these. You can see that in a cell, the cytoskeletal elements are spreaded everywhere. It means that they can support almost every structure of the cell, the organelles, the nucleus, the cell membrane, and the others. And also, they can help in movements of organelles of the cell itself when required. Here is a closer diagram in which you can see that there is a mitochondria inside in a green color. Upper is the cell membrane, and above and below the mitochondria, there are different types of filaments which are attached on one side to the mitochondria, and they are attached on other side to different other filaments, or to the endoplasmic reticulum, or to the cell membrane. Actually, mitochondria are kept in their place with the help of these different types of cytoskeletal elements, some actin filaments, the microfilaments, some microtubules, and some of the microfilaments. Here is another view of the same thing. Here you can again see cell membrane, endoplasmic reticulum. From another view, you can say about roughly surface view, you can see the same thing, the endoplasmic reticulum, the microtubules, the microfilament, and one mitochondria entangled in them. Just like mitochondria, other organelles are also kept in place. They are also kept in place with the help of these cytoskeletal elements. And whenever they need to move, for example, if energy is required, because mitochondria produces energy molecules, If energy is required in another part of cell, then mitochondria will be transported from this part to the other part of cell. And these mitochondria will be moved with the help of the microtubules. Now we finished talking about the cytoskeleton, and now we are going to talk about the cell's organelles, different types of organelles present inside the cell. What are organelles? Organelles are subcellular structures. For example, the mitochondria, as we have seen in the previous diagram, the ribosomes, which makes proteins, the lysosomes, vacuoles, so on. We look at them one by one. First of all, the nucleus. Nucleus is a very important part of the cell. It have the heredity, the genetic material in the form of chromosomes. Nucleus is present usually, for example, in animal cells in the center, but in plant cells, the nucleus is not present in the center because particularly the mature plant cells have a very large vacuole present in the center. Due to that, nucleus is moved towards a side. So in plant cells, nucleus is present towards the side, and in animal cells, mostly it is present inside the center. Nucleus, the structure of the nucleus, if we talk about it, it have a nuclear membrane, it have nucleoplasm, and it have a small structure called nucleolus. And in the nucleoplasm, it have the chromatin material, the heredity material, the DNA, deoxyribonucleic acid. Look at the diagram of the nucleus. If you look at the diagram, you can see an external membrane. This membrane is called the nuclear membrane. After the nuclear membrane, a fluid is present that is called the nucleoplasm. Nucleo of the nucleus, plasm, the fluid. This nucleoplasm have chromatin material inside. You can see a darkly stained body inside, which is called nucleolus. This is a site where ribosomal RNA is produced. If you observe the nuclear membrane, you will find out that there are pores present on the nuclear membrane. These are called nuclear pores. As we know that the nucleus have to exchange materials with the cytoplasm of the cell. These pores are for that transport of materials. For example, if a messenger RNA is produced inside the nucleus, it have to be transported out. It will go out through the nuclear pores. Now we talk about the next organelle, the ribosome. Ribosomes, as you can see in the diagram, these are the organelles which makes proteins. These consist of two subunits, one large, one small. These can read the messenger RNA, which is made by the DNA, the heredity material, and they makes proteins after reading this particular material. They are present in the cytoplasm free or attached to the endoplasmic reticulum. There are a large number of ribosomes present inside the cell. Few cells which are metabolically more active, they may have more ribosomes. Few cells which are metabolically less active, they may have less number of ribosomes. And the eukaryotic ribosomes are slightly larger than the prokaryotic ones. Next comes the mitochondria. Mitochondria are the organelles which are called the powerhouse of the cell. They produces energy required by the cell in the form of ATP. ATP is a molecule, adenosine triphosphate, which have high energy bonding in its side. When it breaks, it releases lots of energy in the chemical forms that cells can use for performing their different functions. Mitochondria are double membraned organelles. They have a double membrane on their surface. The outer membrane is comparatively simple membrane, a straight membrane, and the inner membrane is highly folded. Its folds are called cristae. These membranes are filled with a fluid called the matrix. In this matrix, they have enzymes. Mitochondria have their own DNA, and they perform their many of the functions with the help of their matrix and their inner membrane. Their outer membrane is less specific. The inner membrane is more specific and highly organized. Here in the diagram, you can see a mitochondrion. Mitochondria is plural. Mitochondrion is singular. In the diagram, you can see the outer membrane, less folded, straight. The inner membrane, actually not folded, straight. The inner membrane, highly folded, have cristae and a matrix inside. On the left, you can see an image picture taken through the electron microscope. In this picture, you can see a mitochondria in a grayish color. The next organelles. These are found only in plants, plastids. Plastids are another type of organelles which produces energy, and they produces also carbohydrates. They have another property that they produces carbohydrates. These are also double membrane bound. They also have an outer membrane just like mitochondria, which is straight or smooth, and an inner membrane which consists of highly folded and organized structures called thylakoids. Plastids are present only in plants. They have specific pigments which can absorb sunlight and produce energy for them in form of ATP, and they also can convert inorganic carbon into the organic carbon. That is, they convert carbon dioxide by a series of reactions into the carbohydrates, and these are the carbohydrates which are source of stored energy for all living organisms on earth. These plastids are of three types: chloroplast, chromoplast, and leucoplast. Chloroplasts are green in color. They have a green pigment, and these are the organelles which are responsible for the energy production and the carbohydrate production in the plants. We call them photosynthesis in the Calvin cycle. They do photosynthesis, and they produces energy and carbohydrates. The chromoplasts are other types of plastids which are present in the flowers and the fruits. We observe different beautiful colors of flowers and fruits. These colors are due to the presence of the chromoplasts. Chromoplasts may be yellow, maybe red, maybe more darker in color or in different shades. There are third types of plastids called leucoplasts. These are present usually in the roots and in the tubers. These plastids store different types of materials for the plants. For example, proteins. There are protein leucoplasts have their types. For example, there are proteinoplasts that saves protein. There are other plastids which save starch like amyloplasts and the others. They also have a double membrane, and external is a smooth one, internal is a folded one. Then comes the next organelle, the endoplasmic reticulum. I should say endoplasmic, present inside the plasma. Reticulum, a network. These are a network of interconnected channels. These are present in various parts of cell, particularly surrounding the nucleus. The endoplasmic reticulum is of two types: rough endoplasmic reticulum and a smooth endoplasmic reticulum. The rough endoplasmic reticulum is called rough because on its surface ribosomes are attached and they appear as granules. So it gives a granulated appearance. We call it rough endoplasmic reticulum. Other is called smooth, don't have ribosomes attached to its surface. You can observe these in a diagram. In this diagram, you can see on one side of the nucleus, there are rough endoplasmic reticula present. On the right, you can see the same thing in an electron microscope image. What is the function of the endoplasmic reticula? Basically, ribosomes makes proteins. These proteins enter inside the endoplasmic reticulum. Inside these reticulum, there are many types of enzymes, and these enzymes modify these proteins and convert them into their final shapes. So endoplasmic reticulum has very important role in the modifications of proteins from their basic structure into their final structure. And this role is very important because if even one protein is not formed correctly, the result will be loss of its function. And loss of function may result towards various diseases. Now we talk about the next Golgi apparatus, also sometimes called Golgi complex. Golgi apparatus, we call it Golgi apparatus because it was discovered by the Camillo Golgi, scientist. This is also called the post office of the cell. When proteins are produced, they are modified by endoplasmic reticular enzymes. Then they go outside towards the Golgi complex, the Golgi apparatus. Function of the Golgi apparatus is to pack these proteins and send them to the appropriate place where they are required. Some proteins are secreted outside. Golgi complex pack them into small vesicles we can say fluid-filled packs, which are called secretory vesicles. Some other proteins are packed for sending towards inside of the cell. So Golgi apparatus is the post office of the cell. And as in the diagram, you can see it consists of various flattened discs, and these discs, as the diagram shows, is releasing different types of vesicle on towards various parts of cells. As you can see, few vesicles are going down, few are going up. It means they are going in different directions as required by the cell. This is associated with endoplasmic reticulum because it have to get the materials, the proteins prepared by endoplasmic reticulum, or maybe lipids produced by other type of endoplasmic reticulum, the smooth one, and then pack them and send them different parts of the cell or maybe outside the cell. Next is called centrioles. These are the next organelles. These are present inside the cell near to the nucleus. They are hollow and cylindrical bodies. Two are present near to the nucleus, collectively called centromere. Their function is during cell division. When cell divides, these centrioles move towards the sides of the nucleus and make the spindle fibers. These are present in animal cells. Then comes the vacuoles. Vacuoles are also fluid-filled, single membrane-bound organelles. These are present in different forms. For example, the food vacuoles, the contractile vacuole, and their major function is storage of different types of materials. For example, some proteins. Sometimes cell is producing some waste material, and they have to preserve that waste material for some time and then remove it towards outside. The food vacuoles, particularly in the unicellular organisms like amoeba, when they get a food material, they invaginate it and convert it into a vacuole. Then this food is digested. So vacuoles are also very important in the cells. In animal cells, there are many small vacuoles. In plant cells, there is, in particularly mature plant cells, there is a single large vacuole which is produced by joining of various small vacuoles. Another organelle is called lysosome. Lysosomes are membrane fluid-filled sacs. They have different types of enzymes inside. They are called the organelles for garbage disposal. Whenever anything is produced in the cell, it have to be broken down. When it is broken down, it have to be placed in some time in a particular organelle. Usually vacuoles do so. Lysosomes have enzymes. They fuse with the vacuoles, which have those waste materials, and pour their enzymes inside those vacuoles, and those enzymes break down those waste materials and convert them into less harmful products for the cell, and then they are released out. So lysosomes are the disposal system of cell. Their function is to dispose of various materials which are harmful for the cell. These material may include the bacteria or viruses which invaded the cell and need to be digested. So this is all about various cellular components. Today we talked about the cell theory and the cellular components. I hope what I said you understand it. We will meet in the next lecture. Thank you.